know that Mendel's work on sweet peas has been the single most important pillar of classical genetics. It's been so foundational that these laws still hold today, now accompanied by the expansions of non-Mendelian genetics. Since the genome is an incredibly complex dynamic system, new rules were needed to govern the interactions between different genes, interactions with the micro and macroscopic environment, and parental influence. And this is how non-Mendelian inheritance models came to be, encompassing environmental influences, chemical modifications of DNA, the role of cytoplasmic organelles, and intragenomic interactions. Starting with the strangest case of non-Mendelian inheritance of all, let's talk about mitochondrial inheritance. First things first, mitochondrial inheritance is exclusively maternal, meaning a sick mother will pass the condition to all her children, but only her daughters will pass it on to theirs. On top of that, mitochondria have a unique feature: hundreds of copies of their own DNA per organelle, which means thousands per cell. This means some DNA copies will carry the mutation, while others won't. This condition is called heteroplasmy, and it's been the basis of the threshold effect. For a mitochondrial disease to manifest, the proportion of mutated molecules must reach a minimum threshold. That threshold is typically around, on an average, 80%, the point at which the mitochondria can no longer produce enough energy to keep organs like the heart or brain running properly. Genomic imprinting, on the other hand, describes the silencing of certain genes based purely on their parental origin. To Mendel, this would have seemed absurd, and yet, during gametogenesis, DNA methylation silences specific regions, and well, these modifications are then maintained to the fertilization and throughout the development of the new individual. The most well-known clinical example involves the region from Q11 to Q13 on chromosome 15, which contains several genes, some subjected to maternal imprinting, others to paternal imprinting. Loss of the paternal copy leads to Prader-Willi syndrome, characterized by obesity and hypotonia, while loss of the maternal copy leads to Angelman syndrome, which instead presents with intellectual disability and a distinctive behavioral profile, including frequent laughter and hyperactivity. Now, let's talk about when one gene gets to decide the phenotypic fate of its neighbors. And we are bringing Labradors into the story. These big, lovable dogs have two alleles for coat color, black and chocolate. But they also carry a second gene that controls whether any color ends up in the fur at all. If the second gene is homozygous recessive, no pigment is deposited, and our four-legged friend ends up golden, regardless of what color the first gene was actually coding for. This phenomenon is called epistasis, and, well, the gene responsible is known as the epistatic gene. Now, these are fairly stable dynamics, but there are also conditions caused by unstable genetic defects. We are talking about trinucleotide repeat expansions. During DNA replication, DNA polymerase can slip. During DNA replication, DNA polymerase can slip in certain regions, inserting more and more repeats with each round. Well, this leads to genetic anticipation, meaning that as generations go by, the disease will appear earlier and earlier, and with increasingly severe symptoms. And wait! What happens when a single gene influences many different traits? Some inexplicable coincidences suddenly stop being mysterious. Think about those white cats with those stunning blue eyes. Well, they are generally deaf. Why? That's because the gene controlling pigmentation is the same one that drives the development of the auditory nerves, specifically during the migration of neural crest cells. These very same cells later become melanocytes or cells that line the inner ear and make hearing possible. The phenomenon is called Pleiotropy, when one gene tries to do too many things at once, or simply manages processes that start out close together but ends up in very different places. Oh, and unlike Pleiotropy, a single trait can be shaped by the combined influence of multiple genes. Polygenic traits like height or weight are continuous rather than discrete because, well, they represent the sum of many small efforts coming from different genes. When environmental factors are polygenic and multifactorial traits, since, well, they are now influenced by factors beyond DNA alone. Now, don't say this around, but some genes can decide whether or not to actually express the trait they encode, which would have been heresy to Mendel, but it's simply called incomplete penetrance, the model in which not all carriers express the trait, while in variable expressivity, the same gene can produce different effects each time it's expressed. In polydactyly, the same gene can result in a different number of extra fingers in each individual, while in osteogenesis imperfecta, not all carriers display the mutated phenotype. And of course, just to keep things simple, both effects can be present at the same time. Woo! Okay, we now come to a particular case. The models we are about to discuss sit very close to Mendel's law, but deviate from them on a few key points. In incomplete dominance, the law of segregation and the law of independent assortment are both respected, but the resulting phenotype is an intermediate blend of the two original traits. In codominance, on the other hand, both dominant alleles are fully expressed, but simultaneously, as in the case of the blood type, A, B. Finally, in multiple allelism, more than 2 alleles exist for a given gene, and this can be arranged in a hierarchical dominant scale or give rise to other non-Mendelian inheritance patterns. And this very long catalog is still far from complete. Research keeps pushing forward, and we are still today adding roots to the book. How DNA and RNA really interact. Which is the real power and role of RNA? Is dark DNA